My name is Pia Abola, and I'll be your host for today's webinar. Our speaker is Applied StemCell CEO Ruby Tsai, and the title of her presentation is The Future of Allogeneic Cell Therapy, a deep dive into applied stem cells' hypoimmunogenic donor cells. Dr. Tsai is one of Applied Stem Cell's founders and has been using and developing iPSCs to model disease and enable cell therapy development for over 15 years. Her presentation will take approximately 25 minutes, and then we'll have 5 minutes for questions. Please feel free to add your questions to the chat during the talk, and we will address them at the end of the presentation. And if we don't get a chance to get to your question during the presentation, we will follow up with you via email. So with no further ado, I'd like to hand everything over to Ruby.
Thank you very much, Pierre, for the introduction, and thank you everybody for attending this webinar. So cell therapy is a promising and powerful next generation medicine with the potential to transform treatment across disease indications with significant unmet needs. And so based on the donor cells, cell therapy are classified into two categories. One is autologous, meaning that the cells are taken out from the patient and then manipulated ex vivo and then transplanted back into the patient's body. So that's one product or one cell line per patient. All right, so that's autologous cell therapy. And the other one is allogeneic. So allogeneic cell therapy, starting from donor cells from a healthy donor, and then the cells are manipulated ex vivo, and then the cells can be used to treat many patients. Right? So in this case, one cell line is for many patients. And the current approved cell therapy, for example, for CAR T cell therapy, there are 6 of them that are approved in the US, and they are all fall into this category of autologous cell therapy. And because of the nature of autologous manufacturing, and these cell therapies are very expensive, they're about $400,000 to $500,000 per dose per patient. And usually patients would need a couple of doses. So we're talking about a couple $1 million for a treatment. So they're very expensive. And of course, also takes time to make these cells. So they take time. In some cases, this is a life and death situation, right? So what we want to do is more focused on allogeneic. And furthermore, we wanted to start with iPSC. So iPSC has the property of unlimited, basically divide indefinitely, right? So really provide unlimited starting materials for cell therapy. And then another thing is iPSC can be differentiated into virtually any type of cells in the body. So they can provide really a diverse cell types for many different disease indications. So that's what really what we're focusing on, is really using iPSC as starting cell types, and then do gene editing to manipulate it so that these cells can be therapeutic, right? And in cases here that for this particular topic we're talking about today is really to make it how to manipulate the cells that cells can be evasive, right? Escape body's immune system. And then these cells can be differentiated into specific cell types depending on disease indications that one is studying, right? So it can be neurons, hepatocytes, or T cells and NK cells, any other cell types. And these are products, off-the-shelf products that can be used to treat many patients, right? So this is really a very, this is the goal that we're all trying to achieve, like how we can really revolutionize cell therapy, where it can be used for much broader population and much more affordable cost. So we see this as a very promising route to go after. And so obviously one pain point in allogeneic cell therapy is immunogenicity from donor cells, right? So this just one example of how body's immune system work. And it's very complex. As you can see, this is only part of the genes that are illustrated here. So it's a very complex. So the body's immune system is very smart and complex, and they will reject anything that comes in from outside the body, right? So in order to for the cells to take these donor cells, there are many genes that need to be manipulated so that the donor cells can escape body's immune system. So the main route that the immunity works fall into two main categories. One is really cell mediated immunity, and the other is humoral mediated immunity. And in the cells, it's mainly T cells and K cells, and they're also macrophages and dendritic cells, right? And so if we look at this, so the cell-mediated immunity here is in green boxes, right? So they're mainly CD T cells, and then the humoral immunity, the antibodies, they're produced by B cells, yeah. So, so when we think about how we can make iPSCs universal, right, immunogenic, hypoimmunogenic, and so there are several pathways we really have to look into it, right? So talked about T cells, we talked about NK cells, macrophages, right? So, and there are genes that involved in mediating these immunity. So the way that we could do is to really manipulate these genes so that the body's these immune cells won't recognize the donor cells, right? So there are multiple pathways that we can look at is, for example, HLA class 1, class 2 genes, they mediate T cell immunity, right? And then there are also NK cells, they're receptors on the NK cells. So they are inhibitory receptors that we can block those receptors so that NK cells will not kill the donor cells, right? And then macrophages and their genes that can actually block phagocytosis. So these are the different pathways that we want to manipulate so that the donor cells will be hypoimmunogenic, right? So I mentioned the class 1, class 2 major histocompatibility complex or molecules are the very important provide immune surveillance for T cells. And in humans, MHC are the same as HLA. And so class 1 genes, they contain subunits. And one common thing is that they all interact with B2M, so beta microglobulin. So this is a common factor they all interact with, right? So this is important because, when we talk about strategy, about knocking out class 1 gene, and this is actually a key factor to knock out so that class 1 is no longer expressed. And then there's also class 2, so contains basic heterodimer, right? So you have these 4 subunits and form a complex. And so these are the 2 major genes that provide immune surveillance against T cells. So the strategy, like I said, I already implied that to knock out class 1 is really to knock out B2M gene that codes for the beta microglobulin gene. And so by knocking out this gene, that will block basically destroy the class 1 expression. And this way that would block T cells. And then for class 2, so there's a common factor also here is C2TA. So C2TA encodes a transactivator. So this is a transcription activator here that drives class 2 gene expression. So if C2TA is knocked out, and the expression of class 2 genes no longer happens. So that really shuts down the expression of class 2 HLA expression, right? So these strategies have been used in multiple, publications. I'm just using one of them to show that knocking out class 1 and 2 are protected. The donor cells are protected from T cell response. So, for example, the wild type iPSC is in blue here, and then B2M knockout, single knockouts yellow. So we're measuring really T cell response, and you see much lower response from T cells. And then C2TA knockout, and you also see a lower response. And this is the double knockout, right? So both class 1 and class 2 are knocked out, and then it's even better, right? So really knocking out these two HLA genes pretty confer the donor cells to evade T cells. And however, so this is not the whole story yet. So double knockout, you can convert the donor cells to evade T cells, however, trigger NK cells. So now there's a signal kind of sending out to NK cells says, eat me, all right? So now this activates the NK cell. So NK cells can still kill the donor cells, right? So in order to do that, so there are other strategies to really block the NK cell pathway, right? So there are multiple ways of doing that. So their past, publication has shown that HLA-C, E, G, and F, these gene overexpression, each one of them could help to prevent NK cell killing, right? So I'm just using some examples here and go through the data a little bit quick in the sense of time. So this is HLA-C. So expression of HLA-C can inactivate NK cells, right? So wild type, so we're looking at NK cell activation, right? So this is a wild type control, and this is retaining the C, HLA-C, and this is B2M knockout. This is a positive control, K562 as control. So in the presence of HLA-C, we see a protection of from NK cell killings because you don't see activation of NK cells. And this is an example of HLA-E overexpression, and also so protection from NK cell killing is shown in the expression of HLA-E dimer here, right? So this is a pre expression, and this is after HLA-E expression. So you see a decrease in cell killing. So this is to measure cell lysis, right? So expression of HLA-E dimer really protect the cells from NK cell killing. And HLA-F, so I want to spend just a couple minutes on HLA-F expression, because this molecule has never been, wasn't reported before, but we actually have a patent on this and have data to show that expression of HLA-F also protects the donor cells from NK cell killing. And this is because HLA-F interacts with NK cell inhibitory receptors. Here is just, representative, right? So one of the receptors, LIR1, is an inhibitory receptor for NK cells, and HLA-F binds to this receptor and then inhibit NK cell, right? So we have a patent that covers HLA-F expressed donor cells that protect the cells from NK cell killing. And so the patent covers HLA-F modified cells and methods in cell transplantation studies. And this patent was just issued in September. And one key thing that we used is a membrane-bound fusion strategy, right? So it's HLA-F fused to B2M. And this is because this membrane-bound form interacts specifically with inhibitory receptors on NK cells to inhibit NK cell killing. So this is some data. So this shows HLA-F overexpression in HEC293 cells. This is in iPSCs. So a different ratio between the effector cells and target cells. And we see a decrease in cell death when HLA-F is overexpressed, suggesting that cells that express HLA-F is protected from NK cell killing. So now the last example is HLA-G is also has shown that protect cells. So you just look at a gray bar. These are cells expressing HLA-G compared to the wild type. So those the HLAs are actually mostly for NK cells, right? So now CD47 has been talked about quite a bit, and so it's really made for the protection from macrophage phagocytosis pathway, right? So I'm just going to quickly go through. So this is from Toby Duas's lab, and they showed very elegantly how CD47 expression inhibit phagocytosis. So you look at non-modified versus overexpressed CD47. So in this thing, they also knocked out the class 1, class 2 genes and overexpressed CD47. So they're looking at by ELISpot studies, and then they can see that overexpression of CD47 inhibit phagocytosis. So then in summary, so basically if you can co-express all these genes we talked about, so we should be able to block multiple pathways, right? So so I'm just showing here these different strategies. And in this one example is that knocking out the class 1, class 2, and then you overexpress CD47, HLA-G, and another gene I didn't specifically talk about, PDL1, you see protection from those major pathways, right? So T cell cytotoxicity, T cell killing is reduced, and NK cell cytotoxicity is also reduced. And then phagocytosis, we'll look at this orange line. It has has knock-in, right, of multiple genes, right? And so these pathways all seem to, be blocked. So that really confer much better hypoimmunogenicity to the donor cells, right? So, and there are also other candidate immune genes. And so this is taken out from Andrzej Nagy's lab's paper. And so they also looked at many different genes. So the reason I brought this up is that we also have our own favorite genes, like to really block different pathways. So I will be talking about Serpin B9. So this really to inhibit granzyme B. And we see this is very important because it really blocks apoptosis, right? And then another one is CD200. So this really is kind of similar function to CD47 that blocks phagocytosis. And then another factor that we are going to be using is CCL21. So this has not been used in the previous papers that I just presented. So this is really related to dendritic cells. So we also think it's important if we can confer, either a much better hypoimmunogenicity, and this is to block dendritic cells, right? To disrupt migration of dendritic cells. So our strategy is try to block multiple pathways so that the cells have the best, evasion or clerking function from the body's immunity, right? So that's really our thinking behind making hypoimmunogenic cells, is combination of these multiple pathways. So our approach, basically the idea is very, similar to other people have been thinking about, but we just taking a more comprehensive way, and also keep in mind of what would be the most efficient way to manipulate, right? Because we're talking about many genes and how we can do it most effectively. We don't want to do like 5 rounds of gene manipulation because it's not just time consuming, but also the quality of the donor cells can be compromised from multiple gene manipulation. So here's what we're going to do, right? We're going to start with our cells, which is we call iPSC target masked IPS, the target masked iPSC line. So I will explain a bit more later. So basically, we have a master cell line that has a landing pad inserted in a safe genomic harbor locus. And then we can repeat, insert any genes of interest. Another thing is really the technology itself. Target allows large cargo insertion. We can insert up to 20 kb. And that really means that we can insert multiple genes in a single step, as long as they fit into 20 kb, right? And so this is how we can do it. So basically, we have 2 phases here. The first phase is knockout. So we're going to do knockout of class 1, class 2. So that's very common. People have done that. And the only thing that we do differently is that we will be using Mas7 to do knockout, not CRISPR-Cas9. So it's very obvious, for very obvious reason, is freedom to operate. So we keep that in mind. So because we want to create a cell line that's GMP grade and with freedom to operate so that people can use it, right, without concerning about IP landscape. And then the second phase is really one-step knock-in of multiple genes using our proprietary target gene insertion technology. And so these are the candidate genes that we're going to knock in. So I mentioned HLA-F, which is our patent that can invade NK cells. I mentioned in a previous slide, SerpinB9. It's a proteinase inhibitor that can inhibit glyzyme B. And so that will block both T cell, NK cell, and other cell killing because it really blocks apoptosis, right? And then CCL21 disrupts migration of dendritic cells. They'll block the dendritic cell pathway. And then CD200 would inhibit phagocytosis by monocyte and macrophages, right? And as I mentioned, so we do all these in iPSC because iPSC has the potential of becoming any type of the cells. This really add another level in terms of universal cells, right? And then also provides unlimited starting materials, really for truly off-the-shelf cell therapy product development. So just a few words about TARGATT. So TARGATT is a technology that we licensed exclusively from Stanford University. I am a co-inventor of this technology when I was at Stanford. And so basically, TARGATT is an integrase-based technology. And integrase mediates recombination between 2 attachment sites, ATTP and ATTB. And upon the combination of these sites, and in this case, it results in integration of the transgene here illustrated in green, right? And because this integration is actually unidirectional, so that's why TARGATT works a lot more efficiently in terms of gene integration compared to any other technologies that are on the market. And this is because these 2 recognition sites are not identical. They're totally different sequence. So after integration or recombination, it creates 2 hybrid sites, ATTL and ATTR. So these sites are no longer be recognized by integrase. So there's no reverse reaction happen. So for example, like LOXP, accrete LOXP, LOXP sites are identical. So after integration, you still have the same LOXP site. So the reverse reaction also happens, right? So this is why integration by integrase is a lot more efficient than any other enzymes. So this is the technology that we use. And another thing is really the large cargo. So 20 kb is not really the limit of integrase. It's the limit of our cloning capability on a plasmid. And so we have been successful in inserting 20 kb, and we have published a paper that co-express 4 genes in a row in one cassette, and in one step. And so this is really useful in this case for making hypoimmunogenic cells for co-expressing so many genes in a single step, right? And another thing is that all these steps, so we're making everything in GMP grade so that these cells can be used directly for clinical application. So everything, all processes, testings, all follow GMP requirements, and using CGT grade reagents. And we're preparing a drug master file, and we plan to file with the FDA by January 2025. That's the starting cells, right? And so here's our timeline. So I already talked about strategy, right? We're going to knock out the class 1, class 2 genes. So phase 1 is really knocking out using MAT7. I emphasize MAT7 because CRISPR-Cas9 IP landscape is very complex, so we don't use that for GMP manufacturing. So we use MAT7, and we have done that very successfully. And in GMP. And in fact, that's how we made our target master cell line, is using MAT7. And so this particular step, we aim to finish it by Q1 2025. And our phase 2 will start right after that, and which is knock-in of these multiple genes using our TARGATT technology. And we aim to finish that by Q3 2025. So we're getting very close to really reach our goal to create an ideal hypoimmunogenic iPSC line for people to really use it for cell therapy development. We also welcome collaboration partnership on making customized hypoimmunogenic cells. This because every project or product has its own requirement in terms of either making the hypoimmunogenic cells or actually expressing some therapeutic genes. And we can all do that using our TARGATT technology. So we're really welcoming any other potential collaboration and partnership at this time. So basically, to summarize, we're very close to making the allogeneic cell therapy a reality. So the takeaway message is that really allogeneic is what we all see it is a disruptive technology. And the main hurdle, and we're trying to tackling it, is immunogenicity, right? That's our main hurdle. And so we're doing that. Another thing is really payload. So other approaches using CRISPR, Cas9, or any other methods all require multiple steps. You have to knock in multiple genes in multiple steps. And here, using our TARGATT technology, we can do it in a single step, right? And so our goal is really provide off-the-shelf genetic modified cell line to provide unlimited, consistent starting cells for people who develop allogeneic cell therapy, so that one product can be used for many patients, so it's scalable, and also keep consistent drug product consistency. It's not just because we start from cell banking, but also just because the TARGATT technology, we have this landing pad inserted in a safe harbor locus, so that can be used for repeated insertion. So that really provide another level of consistency, right? So it's kind of like plug and go kind of model. So that really provides another level of consistency in product development. And another thing I didn't get to mention is really gene expression throughout the differentiation process, right? So we see often that gene gets silenced during differentiation process. And that's a big bottleneck too for cell therapy. And we have data to show that our target technology in combination with our locus allows gene expression throughout the whole differentiation process and also internally differentiate the cells. So really ensure that the cells express the genes and functional, right? And another thing is we don't use virus. We don't use lentivirus or retrovirus like all the CAR T cell therapy that's approved or viral-based transfection method. So we use plasmid, which is a lot less costly and faster and safe, right? And so the big thing is I think this is not just cost, it's really life-saving strategy because patients cannot wait, especially cancer patients, right? They don't have time to wait for the cell to be made for in the autologous cases, right? Taking the cells out and being made ex vivo and transplant back. Just simply, they don't have waiting time, right? So, and another thing is by reducing the cost, so the product can be really used for much broader population. So they can really, so cell therapy can be a true really medicine for all people, right? So with that, thank you very much for your attention, and I'll be very happy to answer your questions. I run a bit over time though, so.
Yeah, so thank you very much, Ruby. We do have some questions. I'd like to start with one. How did you determine the safe locus for your target-mediated insertion site?
Yeah, so that's a very key question. So I didn't get to talk about it. So our safe harbor locus is our benchmark locus. We've been using it for over 15 years. So that's a H11 locus. So we identified through a genome scan. So that was over 2 years of work. We also published a paper. So that work was done when I was at Stanford University. And we tested in many different organisms, not just cells, right? So we tested in mice initially, and then we also tested in mini pig and a rabbit, and then the many different cell lines. And so it proved to be safe in organism for, does not affect in development, and also for different cell types, and also ensure high level expression. So the locus is intergenic, so it does not disrupt any internal genes in between two highly expressed genes. That ensures that the locus is in open chromatin structure and more accessible to transcription factors, allows high level gene expression.
Okay, I think we have time for one more question. And this is asked, are your 4 transgenes expressed from the same promoter, or will every transgene be driven by its own promoter?
In the case that in the paper that we published, it was under one promoter, and then the genes are separated by 2A sequences.
Okay, thank you very much. We still have a lot of questions, and I will respond to those by email and follow up, but we are reached the end of time. Thank you very much for participating, and we hope to see you at one of our next webinars. Bye-bye.